Hi, everybody, and welcome back. Today, we're going to be looking at mitosis. And mitosis specifically is looking into the process of creating new cells. And we do this for a couple of reasons. Now, the three main ideas that surround mitosis is that we need to repair, replace, or reproduce a new cell. And often our cells are damaged, or they need to be fixed, or they need to be completely replaced because they're old. And that's essentially what mitosis is for. It's to replace old cells or to repair cells so that we can continue to maintain the tissue. Now, in front of you, we have a diagram of what we call the cell cycle. This is essentially the cycle that every cell will go through before and after it has finished dividing. And it's important to know what is happening to the cell. And so we normally start off with our G0 phase or our resting phase. Essentially, what that is we've freshly divided the cell. It's still young, it's an immature cell. And what we need to allow it to do is it needs to duplicate its organelles, it needs to go through protein synthesis, it needs to grow, and basically it needs to replace all of the structures it may have lost once it finished replicating. So it's a fresh new cell. That cell is going to get a little bit bigger and it's going to enter the S phase, which is where we go through DNA replication. And I'll explain a little bit later why DNA replication is so important when we do interphase. So we go through DNA replication, which is when you make more DNA. And that's important because we need to be able to take those copies of DNA and put them in a new cell. And finally, we enter into the G2 phase, which is the final growth phase. It's where the cell finishes its maturation. The cell is really big now, it's full of cytoplasm and its organelles, and now it's ready to enter the stages of mitosis. Now, this whole starting phase, you will notice at the center of the diagram, is called interphase. So, this whole 3/4 of our cell cycle is spent in interphase. And once we finish interphase, where the cell spends most of its time, we can now enter mitosis. Mitosis is divided into 4 phases, which we will go into now and explain exactly how you identify them and what's the purpose of each. And then we end off with something called cytokinesis, which is essentially separating the cell's cytoplasm. Then the whole cycle starts all over again. We've made 2 cells from one original cell, and this process of mitosis always takes a parent cell and produces 2 identical daughter cells. Now, it's important for us to be able to tell the difference between mitosis and meiosis. They are definitely sister processes and they have a lot in common. However, their products are very, very different. Mitosis is going to take a original parent cell and it wants to make 2 identical copies at the end. And what's really important in mitosis is maintaining the number of chromosomes. Now, in mitosis, we want to go from a diploid cell, which means it has the full set of chromosomes, and we want to take that full set of chromosomes and we want to double the amount of information, which we do in DNA replication. And that ensures that when we eventually half the cell into 2, we will end up with 2 more diploid cells at the very end. And it's really important that we know that these cells are identical. They have the same chromosome number, in other words, they're both diploid. And let's use a numerical value just so that we know what diploid means. Diploid means that you will have a full set of chromosomes. So if this was a human, the parent cell would start off with 46 chromosomes, and the 2 daughter cells would end with each having 46 chromosomes in each. So you're maintaining the amount of chromosomes in mitosis. Now, on the other hand, meiosis is still when we take and we start off with a diploid cell. But what we're going to do is we're going to go through a process twice. And what we're eventually going to end up with is haploid cells. Haploid means half. They have half the sets of chromosomes that we originally started with. So if we use the number for this, if humans have 46 chromosomes, the parent cell will have 46. However, at the end of meiosis, you would end up with 23. Now, this makes sense simply because the point of meiosis is to make reproduction cells like sex cells, our ovum and our sperm. And essentially, if you take one sperm cell and one egg cell and you combine their 23 with each other, which you do in fertilization, you will end up with the original 46. So, mitosis and meiosis are very similar. However, their products are very different. Now, before we go into the phases of mitosis, we need to quickly revise what a chromosome is and its structure. And so, chromosomes only exist in a cell like the one you see in front of you when a cell is going through mitosis. When a cell is not going through mitosis, the chromosomes are not as condensed like you see them in the picture. And if I were to just quickly sketch out a cell, that's its nucleus. Sitting inside the nucleus would be this long string like structure. And that long string like structure is called the chromatin network. It's the DNA, it's one long continuous string. That, however, is going to condense and it's going to form these structures, which we call chromosomes. Now, firstly, it's important to note that when chromosomes first appear inside of the cell, they are single strands. Unlike the photograph that I have here, this one over here, this is double. And how that happens is through DNA replication. And so when we started off, we have one chromosome. Then we double the information, which means we take that single strand and we double it. And we stick it to its identical partner. So now there are two sides of the same chromosome. And so what you end up having is one arm is identical to the other arm. Now, this is where a lot of confusion comes in is, well, then is it a single chromosome or is this two chromosomes? So it's really important to know the structure and understand what's happened here. Because we have gone from one strand to two, doesn't mean we have doubled the amount of chromosomes. Instead, what we have done is we've taken one chromosome and we've turned it into two identical chromatids. And essentially, that is because one side of the chromosome is identical to the other. Attaching our chromosome together in the middle is a centromere, and that holds the two chromatids together. And so, when we refer to this doubled chromosome, we refer to it as one chromosome. Eventually, this is going to get pulled apart, which means we're going to take this doubled structure and we will end up with two single individual structures. Now, the moment we pull them apart like that, they're back to being called chromosomes and they're not chromatids. So that means I have one chromosome here. I still have one chromosome in this picture, but when I pull it apart and they move into opposite cells, I now have two separate chromosomes. This is often where a lot of students find difficulty in understanding the chromosome structure and the numbers. And so, maybe to make it a little bit easier, let's work with actual numbers. Let's work with a human cell. If a human cell has 46 chromosomes and it undergoes DNA replication, it will still have 46 chromosomes. The difference is it went from 46 single strands to 46 doubled strands. Now, the fact that they are doubled doesn't change the amount that we have because they're identical. However, we may have 46 chromosomes, but we have 92 chromatids, which refers to these individual arms. So let's begin the first phase that will introduce us to the rest of mitosis, and that is interphase. Interphase is the phase in which a cell spends about 90% of its time. And the purpose of interphase is allowing the cell to grow and mature and to prepare for the next cellular division. And so, because the cell spends most of its time in this phase, this is the most common appearance of what cells will look like. Now, during interphase, DNA replication is going to take place. And I explained this previously, but the reason for that is we want to take this chromatin network that you can see here, and we want to take that whole long string and we want to double that amount. We want to have twice the amount. And the reason for that is we're going to take all that DNA and then we're going to share it into two cells. And we must double the information so we can keep the number the same. Because remember, if we half everything, we're going to lose our information. We want to keep it exactly the same as it was before. Now, when we look at a picture and we're not sure what phase it is because there's no labels maybe, or you have to identify it in a test, we have to look for a couple of things. So this is how you identify it. First of all, you are going to see the chromatin network is still visible. It will have this long spaghetti-like structure on the inside. Your centrioles, which are these structures that are located over here, are still grouped together. And if you remember, these are the structures that are going to actually pull the whole cell apart. And finally, the nuclear membrane is still visible. And that's really important because once the nuclear membrane starts to dissolve and disappear, we can safely say that we have started mitosis. So to start, the first official phase of mitosis is prophase. And essentially, prophase is the very early stages of our splitting of the cell. And the purpose of our prophase is to first of all arrange the chromosomes. Now, the chromosomes come in pairs. And that means, for example, in humans, we do have 46 chromosomes, but they are in pairs. And so that means we have 23 pairs in total. Why do we have pairs? Because you got 23 from your mom, 23 from your dad, and those 23 are complementary to each other. And so they pair up with one another to make your full 46. And so right now, what we're doing is we are arranging them in those pairs to make sure that when we pull the whole cell apart, everybody gets one of each. And so we have a full 46 at the end. Another reason why we have prophase is because we want the spindle fibers to develop. And you can see that happening here at the end of the centrioles. They are busy growing their spindle fibers so that we can pull the cell apart. Now, how do we identify prophase? We identify prophase by looking for a couple of key changes. First of all, you will notice, if you have a look at this diagram, that the membrane, the nuclear membrane, is starting to disappear. In some pictures, it will be completely gone. In others, they will draw this dotted line. The second thing that you are going to look for is that the centrioles have moved to the opposite poles. In other words, they've moved to the north and the south, or the east and the west of the cell. It's important to remember that sometimes the cell picture or diagram that they have given you may be on its side, or it may be from north to south. In this instance, our centrioles are on the east and west of the cell, but in some other pictures, they may have put them on the north and south. They're both correct. And the final, most defining thing about prophase is that chromosomes are now finally visible. In interphase, they're not visible. They're a long string-like structure. But in prophase, they are defined chromosomes like you can see here in this diagram. Now let's look into metaphase. This now follows on from prophase. And there's some definite changes that have occurred in metaphase. And often metaphase is one of the easiest phases to identify because it's so distinctive and they're very distinctive structures that we can look for. So what is the purpose of metaphase? The purpose of metaphase is to ensure that each new daughter cell is going to get one of each of the newly replicated chromosomes. In other words, if we remember that our chromosomes at this point are double-stranded, and so they have two arms, we need to make sure that we're going to pull those apart correctly. And in doing so, and in lining up the chromosomes like you see in the diagram here, it's going to make sure that one strand is going to end up in each of the final resultant cells, essentially making sure that we maintain the chromosome number. And lastly, we need to allow the spindle fibers to attach. And in this diagram alongside, you can see now here that our spindle fibers have grown out of our centrioles and they've now attached to the chromosomes. Now, how do we identify metaphase? We are going to look for the most defining quality, which is the fact that the chromosomes have lined up at the equator. And we can see this here, that they have done this long line down the center of the cell. And remember, like I said to you before, it just depends how they've orientated the diagram that they give you. Sometimes the equator will now just go, instead of from up to down, it'll go from left to right. But they will all be in single file. And that's the most important thing to look for. Now, the second thing that we can look for is that the spindle fibers have attached, but they haven't pulled anything apart yet. So that's really key. They've attached, but nothing has moved or changed just yet. Next, we look at anaphase. Now, anaphase is also quite a defined phase, and there is a really important thing that's happening in this phase. And the purpose of our phase is to separate these sister chromatids. Now, the reason why we want to separate these sister chromatids is because we want to maintain the chromosome number. If we don't separate the chromatids from each other, which remember, that's when we have our double-stranded structure. If we don't separate it and they don't become two single strands in opposite cells, we are now going to have a cell with too many chromosomes inside of it. So it's really important that we pull them apart correctly. Now, how do we identify anaphase? Anaphase has two important things to look for. Firstly, we need to look for the spindle fibers shortening, which you can see here that they have pulled back. They've gotten a lot shorter than they were before. And as they pull back, they are going to be pulling apart our sister chromatids. And then the second thing that we look for is that the cytoplasm starts to sink in. Now, it's very, very subtle. It's not always this defined like it is in this picture over here, but it's going to sink in more and more over time. And that's because anaphase is then slowly going to fade into the next phase, which is telophase. Last but not least is the phase of telophase. Now, telophase sometimes is drawn slightly differently. So this picture here has what we would call slightly earlier telophase, but I'll show you what it can also look like. And the purpose of telophase is to separate our cytoplasm. So you will notice at the very end here now that it's starting to look like two very distinct cells. Another reason why telophase occurs is that we need to reform the nucleus. We've just freshly pulled apart those chromatids, which are now no longer called chromatids. That's very important. They are called chromosomes. Chromatids only exist if they're attached by a centromere to another chromatid. Otherwise, we call them chromosomes. And it's important for us to reform the nucleus around these chromosomes because we want to protect them and we want them to go back to being in a chromatin network. Now, how do we identify telophase? We are going to be looking for a nuclear membrane that is reforming. And if you have a look in the diagram alongside here, you can see that that is happening. It's that little squiggly line that's slowly fading back in. Then we're also looking for the fact that these are single-stranded chromosomes. So they were originally duplicated. We've now pulled them apart and now they are going back to being a single strand. One strand has moved into one daughter cell, the other strand has moved into the other, still maintaining the number of chromosomes. And finally, we have something called pinching in of the cytoplasm, which if you have a look at the diagram, you can see right here in the middle, it's much more defined than it was before. Now, at the end of telophase, we go into sort of a subsection or a subphase called cytokinesis. Cytokinesis is essentially the separation of the cytoplasm, and it creates something called cell cleavage. And the cell cleavage is literally what I was talking about just now, where there's this little pinching in bit. And that is where we pinch the cytoplasm off, the membrane closes off, and we end up with two distinct cells. Now, if they give you telophase a little bit later on, or even perhaps at the end of cytokinesis, you won't always see the diagram like the one I have here. Sometimes they make telophase two separate cells already, or two cells quite close to one another and brushed up against each other. So sometimes this would be early telophase, the first diagram, whereas this second diagram is more later telophase at the end of cytokinesis. So let's do a terminology recap. At the very beginning, we spoke about diploid and haploid. This refers to the chromosome number a cell will have. Now, our somatic cells, our body cells, have a diploid amount of chromosomes. In other words, they have a full set of chromosomes. Haploid cells, on the other hand, have half. And these are our sex cells or our gametes, our egg cells and our sperm cells. We then learnt about the two kinds of cellular division, mitosis and meiosis. They are very similar in that they share some phases with one another. However, their products are very different. Mitosis is going to take one cell and it's going to form two identical cells. Meiosis, on the other hand, takes one cell and it creates four unidentical cells. The reason for the different products is because of their functions are different. Then we went into looking at the cell cycle and its phases, and we started off with interphase. Remember, this is the phase where the cell spends the majority of its time, it grows, it matures, it gets bigger, and it prepares for the next cellular division. After that, we went and looked at mitosis, and there were our four main phases, which is prophase, metaphase, anaphase, and telophase. You can be asked to draw these phases, label them, or how did you identify them and what's going on in each of these phases. We also looked at cytokinesis, which is the separation of the cytoplasm. It's sort of the final phase of mitosis. It's not one of the phases. However, it's the final stage or the final process that must occur in order to make a new cell. And we also learned about the cell cleavage, which is where the cytoplasm pinches off and we go from being one cell to two separate cells. I hope that this video was helpful and I will see you all next time. Bye.